Hello students, and welcome to our third recorded lecture on proteins. In this lecture, I'm going to be talking about the levels of protein structure: primary, secondary, tertiary, and quaternary protein structure. So we're going to be starting with the primary structure of a protein. So I hope that you remember what we've covered already. We've seen that amino acids are the building blocks of protein, and I hope that you are becoming very familiar with general amino acid structure and the 20 different R groups that give us our 20 common amino acids. I hope you're also getting used to using the abbreviations for each of those 20 amino acids, the 3-letter abbreviations and the 1-letter abbreviations, a good and very useful thing to learn. But I hope that you're remembering the different groupings that can be made of amino acids, whether they're neutral or polar, whether they're acidic or basic, and so on. So we've seen how amino acids are joined into long chains by peptide bonds. So peptide bond here, here, and here between each amino acid. Okay, so here is a polypeptide chain or a polypeptide, or as it's only 50 amino acids, I think we could still call it an oligopeptide. And we've seen how within that polypeptide chain, there is a sequence of amino acids. Okay, at one end, we have an amino end, an amino terminal end, or an N-terminal end, taken from the N of the amino group. And at the other end, we have a carboxyl terminal end, or a C-terminal end, taking from the C of the carboxyl group. So a linear sequence of amino acids read from the N-terminal end to the C-terminal end. So a direction in the direction of amino acids. So please do ask any questions. I've not had very many questions yet on the forum, so I would love to see some on any of the things we've covered. So please do remember to ask questions. And I think you're going to have a chance this week as well to ask questions live in some meetings with Dr. Mallet. Okay, so we're moving on to think of whole proteins now. And I really want you to appreciate just how many different proteins there are. There are millions and millions of different proteins. If we think of all the different organisms that have slightly different forms of similar proteins, there are many, many different proteins. Here are just a few examples of proteins that you might be familiar with, just being compared in size and shape with one another. So here, for instance, we have quite a small protein, which is lysozyme. And here's another small one, which is insulin. Here we have a very large protein, aspartate transcarbamylase. And another fairly large one is catalase. Here is a long, thin protein, collagen. We're going to be thinking a little bit more about that next time. And here is a protein with an interesting shape. It's got holes in. This is a porin, a protein that allows molecules to pass through cell membranes. Here is something long and thin, which is not a protein. This is DNA. It's put into this diagram because it's interacting with deoxyribonuclease, but please don't think that this means that DNA is a protein. DNA is a nucleic acid. Its building blocks are nucleotides, not amino acids. Okay, so DNA is not a protein, but all these other things are proteins, many, many, many different sizes and shapes and functions. Two on here that I want you to particularly notice are hemoglobin and myoglobin, because we're going to be talking a lot more about those soon. Okay, so we've seen that there are 20 different amino acids, but thousands and millions of different proteins. So how do those 20 amino acids give us so many different proteins? That's what we're going on to look at next. So every amino acid has a different amino acid sequence. So here we see an amino acid sequence: proline, alanine, asparagine, lysine, threonine, and so on. Amino acid residues join together by peptide bonds into a polypeptide. So this is the primary structure, or part of the primary structure of the protein, the sequence of amino acids within that chain. And because of the particular amino acids in this chain, the protein can fold up into different levels of structure. So here we see secondary structure. This is an alpha helix. Then tertiary structure, a number of different alpha helices and other parts of the structure. And then quaternary structure, where different subunits of a protein come together to form a complete protein. Okay, so the shape of the complete final protein depends on the amino acid sequence of the primary structure. It's the sequence of amino acids, the order that these amino acids come in and which particular amino acids are there, that give the protein the ability to fold up into these levels of structure and form its final shape. So 4 different levels, as I've already said: primary, secondary, tertiary, and quaternary structure. And I'm going to be talking about all of those today, starting with primary structure. Okay, so the primary structure of a protein describes the way that amino acids are linked by covalent bonds. So the peptide bonds, as we've already seen, are covalent bonds in the protein molecule. There's also another type of covalent bond which is important in proteins, which is disulfide bonds. So if we're describing the primary structure of a protein, we need to give several different sorts of information. So these could include the number of each amino acids the protein contains. So this is just a short stretch of a protein, but in here you can see that some amino acids are present several times. So for instance, we've got alanine here, and also here. We've got lysine here, and here, and here, and so on. So we might use this to say that there is 1 proline, but 3 lysines. Okay, so the number of amino acids is important. So proline is 1, alanine is 3. Different proteins can also contain more than 1 polypeptide chain. So to describe the primary structure, we have to say whether we're talking about the primary structure of just one of those polypeptide chains, or talking about several different polypeptide chains. So we have to know how many polypeptide chains the protein has. It's also very useful to know the N-terminal and C-terminal amino acids of each of those polypeptide chains. This is very helpful in protein sequencing, which we'll talk about later. And then to really fully describe a primary structure, we describe the sequence of amino acids in each polypeptide chain. So here we'd have proline, alanine, asparagine, lysine, threonine, and so on. Okay, so the primary structure of a protein also includes the number and position of disulfide bonds, the second sort of covalent bonds that we find within a protein. So a disulfide bond, it is made from two cysteine residues, which are able to form a covalent link with each other, which are sometimes called sulfur bridges. And these links can be either inside a single polypeptide chain or between two different polypeptide chains. So here is the way that we can form cysteine from two cysteine residues. Okay, so here is cysteine amino acid. Here's its alpha carbon, and here's its R group ending in a sulfur, a sulfide group SH. And here is a second one. So these two are able to interact with one another to release the two hydrogens, okay? So two hydrogen protons and two negative electrons are released when this bond forms, and a bridge is formed between the two sulfur molecules. This bond is often called a sulfur bridge. You can see that the sulfurs bridge between the two cysteine amino acid residues. And this structure, this dipeptide, well, no, sorry, it isn't actually a dipeptide. It's two amino acids joined together, but not through a peptide bond. This joining of these two amino acids into one molecule becomes a molecule called cysteine. Notice we have a slightly different spelling: cysteine, EI, cysteine, just the I, no E in it. So this is an oxidation reaction. So the two cysteines oxidize together to form the cysteine dimer. And the covalent bond is a disulfide bond, sometimes called a sulfur bridge. Okay, so here is an example of a protein for you. This is insulin. And here the disulfide bonds are highlighted in yellow. And as you can see, insulin has two polypeptide chains, a small chain and a longer chain. Okay, this one's the smaller one, this one's the longer chain, the alpha chain and the beta chain. And there are three disulfide bonds in insulin. There is one that joins two cysteines within the alpha chain, okay, and two joining the alpha and the beta chains to each other. So these sulfur bridges are very important in allowing insulin to form its correct structure, which allows it to carry out its correct function. I hope you know that insulin is an important hormone. It's important in regulating sugar processing in the body. It's what's missing in diabetic people. Okay, so here is insulin. And here are the disulfide bonds. Okay, so to summarize, to describe the primary structure of a protein, it's useful to know the number of each amino acid present, the number of polypeptide chains, the N and C terminal amino acid in each of those chains, the sequence of amino acids in each chain, and the number and position of sulfur bridges or disulfide bonds. Okay, quick pause for questions. So how do we find the primary structure of a polypeptide? Well, this is a diagram from the Leninger textbook that you might want to study a bit more carefully. And it shows the overall scheme that can be used to find the primary structure of a protein. So I'm going to go through each of these steps in turn and talk about them in detail, but I want you to look at just the overall scheme. So here we have a polypeptide. You can see that there's an internal sulfur bridge between two parts of a polypeptide. And we want to find out the primary structure of this polypeptide. Okay, so these are the different steps of the procedure that we can go through to find it out. So first of all, hydrolysis. So hydrolysis means breaking apart the individual amino acids in this polypeptide chain. You remember that polypeptide formation of the peptide bond is a condensation reaction where water is released. Hydrolysis is the opposite reaction where water can be added back in between each of those amino acid residues to return them to being free amino acids, okay? Amino acids free in solution. And hydrolysis can be done by using a strong acid, which attacks the peptide bond and returns the water. Okay, so 6 molar hydrochloric acid will hydrolyze most proteins. So if you take some of a protein and put it in a very acidic solution, it will fall apart. The peptide bonds will become hydrolyzed, and then you can separate the amino acids and identify which amino acids are present and how many of each of them are present. This can be done by various forms of chromatography. That's a separation method. You'll be learning more about chromatography quite soon. But using this method, it allows the number of each amino acid present to be determined. Okay, so we've found how many of each of the amino acids there are. So here they are, just listed in alphabetical order, actually. And the number of each of those amino acids that's present is given. So there's 5 A's, 2 C's, 4 D's, and so on. Okay, so hydrolysis tells us which amino acids are present and how many of them there are, but it doesn't tell us anything about the order that they are because we've broken them all apart. We've broken all the covalent bonds, all the peptide bonds, and also the acid breaks the sulfur bridges. Okay, so the second step, going back to our original protein, because we've destroyed the first sample we had with hydrolysis, going back to a sample of the original protein, the next thing we want to do is to find the first and last amino acid. So this can be done by reacting with a label that binds to one end and then carrying out hydrolysis again. So then we will see the label, which is called FDNB, that will be attached to the amino acid at one end, at the N-terminal end, but not to any of the other amino acids. Okay, so this is how we do this. So FDNB stands for 1-fluoro-2,4-dinitrobenzene. Okay, this is what it looks like shown in red here. So here is our polypeptide chain, and here is at the N-terminal end. So the label attaches, it has a chemical reaction which attaches it to the nitrogen of the amino group. Okay, so there is now a label attached to the amino group of the N-terminal amino acid, but not to any of the other amino acids in the chain. So now we can carry out hydrolysis again, 6 molar hydrogen chloride again, but this time we find that one amino acid has the label attached to it and all the other amino acids don't have the label. Okay, so by using this, we can find out which amino acid is the N-terminal amino acid. Similar but differently, we can also find the C-terminal amino acid. Move down, okay. By cutting it off and identifying it. So to do this, we use an enzyme called carboxypeptidase, which cuts the peptide bond at the C-terminal end of an amino acid chain, of a polypeptide chain. Okay, so here is the C-terminal end of the chain, here is the C-terminal amino acid, and here is the peptide bond which joins it to the amino acid before it in the chain. So using the enzyme carboxypeptidase, which also again adds water into the reaction, we can cut off the C-terminal amino acid, leaving the polypeptide chain one amino acid shorter, and then this can be identified again by chromatography. Okay, so we can find the N-terminal and C-terminal ends of a polypeptide chain. Next step is to reduce any disulfide bonds. So this is done by using a reducing agent to break apart the bond and return the cysteine dimer to two separate cysteine residues. Okay, so disulfide bonds can be broken by using a reducing agent. We said that it's an oxidation reaction that forms the disulfide bond. So to break that bond, we need a reduction reaction. And a good reducing agent that's often used for this purpose is called beta-mercaptoethanol. Beta-mercaptoethanol. So here, the disulfide bond forming cysteine is reduced to two separate cysteine residues. Okay, so this makes sure that the polypeptide chain is unfolded. So the next thing we can do, here's our polypeptide chain with its reduced disulfide bond. We can cut it into shorter peptides and then find the amino acid sequence within each of these peptides. So here we're using two different enzymes which cut the polypeptide chain in slightly different places. Okay, so trypsin cuts it into 4 different fragments here called T for trypsin 1, 2, 3, and 4. And then we can find out the sequence within each of those short pieces of polypeptide using Edman degradation. I'm going to talk about that in a minute. Okay. Here we have cut the chain with a slightly different method using cyanogen bromide. This is a chemical reaction rather than an enzyme. And cyanogen bromide separates it into 3 different fragments, okay? A longer one and 2 shorter ones called C1, C2, and C3. Okay, so now we've got this 2 lots of sets of information, sequences of oligopeptides after treatment by 2 different methods. So what we have now is, if you like, a puzzle. Oh, sorry. Before I'm going to talk about that, I just wanted to show you different things that can be used to cut polypeptide chains. So in the diagram there, we saw trypsin and cyanogen and bromide. And this table from Lenin just shows you where it cuts. So trypsin cuts at the C-terminal side of lysine and arginine residues. So I hope you're remembering what lysine and arginine have in common. They are both basic amino acids. So trypsin, the enzyme trypsin, it's a digestive enzyme. It can recognize those 2 amino acids and cut the C-terminal side of each of those amino acids. Cyanogen bromide carries out a chemical reaction which breaks the polypeptide bond on the C-terminal side of methionine residues. And here you can see that there are a number of other enzymes that cut at different places. So this protease cuts at the C-terminal side of arginines, chymotrypsin after phenylalanine, tryptophan, and tyrosine. What do these 3 amino acids have in common? I hope you're remembering, and so on. So we have a number of different ways of cutting up a polypeptide chain to produce smaller fragments. Okay, so now this is what I was talking about. This is where we have to do a puzzle. It's a bit like doing a word game or a crossword puzzle or something. But we have to work out how these 2 sets of fragments can be lined up using the information we have to give us the completed sequence. So we know the amino-terminal amino acid. We've done the experiment, we've labeled it with FNDB, and we know that the N-terminal amino acid is an E, glutamate, okay? So E has to be at the N-terminal end. And if you look in both of the sets of fragments, we have 1 fragment which begins with an E. So we have to place that at the N-terminal end of the sequence. Okay, so the fragment beginning with E in the T set is shorter than the one beginning with E in the C set. So we have an overlap here. If you see, this ends in PIDPR, which is a sequence we see before the end of this fragment, PIDPR. Then the next amino acids are GASM. So if we look, we can see another fragment here that begins with GASM. So we can place that overlapping. Okay, do you see that? and that gives us a few more amino acids past the end of fragment 1 of the C set. So the next ones are ALIK. So if we look here, we can see that C3 begins with ALIK. Okay, and again, by using that one, that gives us the next few, YLIA, and so on, ending in M, which we see here, YLIK up to M. So that's what we've got here, and then now a small overlap with the TK from here, telling us that this one, TK from here, comes next, and then the last fragment in this set going on to the D, the aspartic acid, that finishes the fragments in both cases. Okay, so that's how you solve the puzzle, if you like. That's how you assemble the fragments and put them back together. So the reason for breaking it up like this is because for a long time we were only able to find sequences of quite short polypeptide chains, up to a maximum of about 20 amino acids long. Okay, so that's the reason for cutting it up. We can't go to the whole full length chain and just find the sequence out, because the information becomes blurry. We get too much uncertainty as we go further down the chain. So here's the overall picture again. Do make sure that you understand the procedure, what's going on, how we use the different parts of information to find the sequence of the whole long chain. And of course, here we only have quite a short polypeptide chain, but this same procedure can be used for long polypeptides, long, hundreds and hundreds of amino acids long. Okay, any questions on that? Okay, so how do we actually find out the sequence of amino acids within each of those short peptide fragments? So this is protein sequencing by a method called Edman degradation, and it's somehow similar to the method we use to find the N-terminal end, in that it labels the N-terminal amino acid. But by using these chemical reactions that were formulated by a man called Edman, which is why it's named after him, Edman degradation, not only the N-terminal amino acids can be found, but a series of N-terminal amino acids. So the label becomes attached to the amino acid at the N-terminal end, and then the peptide bond is cleaved to release that final amino acid. Okay, and the amino acid that has the label attached to it can be identified. And then the procedure can be repeated with the next amino acid. So another amino acid is labeled and another amino acid is identified. And it's because this is a series of chemical reactions, and if it's repeated too many times, the results become less clear and become fuzzy, and it stops being always the 10th amino acid. Sometimes it's the 9th and sometimes it's the 11th, and the results become unclear. But up to a few amino acids, up to about 20 amino acids, we can find out a sequence using this method of Edman degradation. Okay, so whole peptides up to up from 0 to 50 amino acids can be sequenced using Edman degradation. Okay, so originally doing those chemical reactions had to be done separately by hand. But then a machine was invented that can carry out these sequencing reactions very accurately and a lot more quickly than a scientist can in a lab doing it by themselves. You can see here that there are a lot of chemical reagents and there are a number of pumps and a mechanism in this machine which allows the right reagents to be added time after time, and then the released labeled amino acid to be identified each time, and a printout of results will be produced. So this automatic machine is called a sequenator, and it can carry out protein sequencing quite accurately and relatively quickly. So we looked at insulin before. Here is the insulin molecule again. You can see that it's quite a small molecule. It's got 51 amino acids in 2 polypeptide chains. And it was the very first amino acid, the very first protein, to be sequenced. This work was done by a scientist called Frederick Sanger. It was published in 1953, and it had taken him 10 years to work out that sequence, okay? Doing it by hand, 1 amino acid at a time, learning and developing all the techniques that were needed for sequencing. Much longer, much larger polypeptides can be sequenced in a few days using one of those sequenator machines. So polypeptides or peptides up to hundreds of amino acids long can be sequenced in a few days. So that's a very big difference, a big improvement on the 10 years. However, it's now quite unusual to try to directly sequence a protein at all, because a more useful modern approach is to actually sequence messenger RNA, which carries the instructions to make that protein. Okay, if you don't already understand how this works, you'll understand a lot more after the course we'll do on molecular biology next semester. But basically, the instructions to make a protein, each amino acid, are carried in a sequence of nucleotides in messenger RNA. And finding the sequence of messenger RNA is much quicker, much easier, much cheaper than finding protein sequence. So if we find out the RNA instructions, we can find out the sequence of a protein which is made from those instructions in a few hours. Okay, so protein sequences. We know a lot of protein sequences now. And these sequences are stored in data banks. So thousands of proteins from many, many different organisms have been sequenced, and these sequences are stored in data banks which can be accessed over the internet. So these are just the names of a few of these data banks. So PDB, Protein Data Bank, is the one in America. Here's the Worldwide Protein Data Bank, and there's a European Protein Data Bank and another one called UniProt. And these now are all linked with each other, so the same information tends to be accessible through all of them, with slightly different ways of getting to it and slightly different things that you can do with the information depending which one you've chosen. So these data banks are freely available to anyone. Anyone can go to the data banks, look at the information there, use it in their research in whatever way that they need to. So one very usual thing that people do using protein sequences is to compare the sequence of proteins which can perform similar functions in different organisms, or proteins that are slightly different proteins performing a similar function in the same organism. Okay, so an example here, again taken from Leninger textbook, is 3 proteins which are going to be meeting again later: myoglobin, hemoglobin alpha, and hemoglobin beta. Okay, and here are the sequences written out from the N-terminal end here to the C-terminal end here. So I know this is rather small, so I've just made the top part of it here a bit bigger. So you can see here that we're using the single letter abbreviations for the amino acids. And when you're making this sort of sequence, it's very useful to just have those single letters, which is one of the reasons why we use them. And you can see here that a comparison has been made between the amino acid sequences in these 3 similar but slightly different proteins. You'll be learning more about them in a couple of lectures' time. So you can see that the N-terminal amino acid in all 3 of these polypeptide chains is V, a valine. And then you can see a number of other places where the same amino acid is found in all of them: valine here, tryptophan here, lysine here, valine again, and so on. So this sort of comparison of polypeptide chains helps to know which are the conserved amino acids, which are the amino acids which are the same in these different proteins. So for instance, we could be comparing proteins from a human, a rat, and a yeast, something like that, and we could find out which parts of that protein are very important for the function of a protein and therefore don't change, and which are less important for the function of a protein and are likely to change a lot between 3 different organisms, like a yeast and a human and a rat. Okay, so you can look at that in the textbook and see more details about these comparisons in the textbook. So if you want to go to some of these protein data banks and explore, and I'm going to be encouraging you to do that later, here are just free web addresses you can go through. Worldwide Protein Data Bank is www.wwpdb.org. The European Data Bank is held at EBI, European Bioinformatic Institute, with a UK web address, and the American PDB, the address here, rcsb.org.pdb. Okay, so you can go to those and explore, and there's a lot of information that you can find out about proteins in those protein data banks. Okay, a pause for questions. Okay, so on the LMS, I've put a link to this page that you can download and print out if you want, or you could probably just use it on screen if you wanted. So this is an exercise to help you practice a number of different elements of protein sequence that we've been talking about. It's going through the process of finding out the primary structure of a protein, sort of as a paper exercise, if you like. So what I've asked it to be done is that one person makes up an amino acid sequence, practicing using the single letter abbreviations. And I've put a number of rules that should make it all work. Okay, so you can use, actually I've said 3-letter abbreviations here, so you can practice using the 3-letter abbreviations to write an amino acid sequence. Then I want you to look at that amino acid sequence and think about the procedure for sequencing that we talked about. So the first step is to hydrolyze the polypeptide and find out how many of each amino acids there are. So here is a table, but I'd like you to fill in with the information from the polypeptide you've made up. So if, for instance, you'd got 5 A's in it, you'd write 5 next to A here. And if you use a 3-letter abbreviation here and have to use the 1-letter abbreviations here, that will help you to practice relating the 1-letter and the 3-letter abbreviations to each other. So this would be the results of hydrolysis of your protein. Here I've asked you what the N-terminal and C-terminal amino acids are. So the amino acids that you've put at the 2 ends. So that's the information that you get using FDNB or carboxypeptidase. And then imagine that you're digesting your sequence with trypsin and then cyanogen bromide. Okay, so you'll have to remember where the those different things cleave the polypeptide chain and work out what fragments you'd see. I've suggested that you might have up to 5 fragments. Okay, and the same here for cyanogen bromide. So what would the sequence be for each of those proteins, for each of those peptides, sorry. So then you'll put all this information in, then fold over your paper, so this part is hidden, the actual sequence, and swap with a partner. Okay, and if you're imaginative and you're being distanced and separate from other people, you can probably do it by taking a photograph or something and sending it to your friend. Okay, and then the friend will do the puzzle part and work out what the original sequence must have been using the information from the sequencing experiments here. Okay, so hopefully that will be quite a fun exercise. It will help you to practice a number of different parts of protein sequence, and it'll be something that you can do with a partner. And if you've got several friends, you can swap them around between you and have a go with each other's sequencing. Okay, so that's just a little exercise for you to do. I'm now going to move on to thinking about the higher levels of protein structure. That was primary structure. Now we're going to be thinking about secondary, tertiary, and quaternary protein structure. So back to this diagram, which I keep showing you. So here is our primary structure, which we've just talked about. So the first higher level is secondary structure. And there are a number of different types of secondary structure, and I'll be talking about a couple of them which are illustrated here: alpha helix and beta pleated sheet. Okay, and then there is also tertiary structure, where those elements of secondary structure fold up on themselves, produce a third level of folding. And then the fourth level of structure, quaternary structure, is where different subunits come together to make a whole protein. Okay, so we'll be going through those levels of structure and seeing exactly what's happening at each stage. Okay, so back to what we know already. We've seen this polypeptide chain before, with the peptide bonds not being able to fold, but rotation and folding being possible on the bonds on either side of the alpha carbon. And we've seen the way that the R groups stick out on opposite sides of the chain, okay, on alternating sides of the chain. So the question is, how does this polypeptide, which we're seeing here just in a long line format, how does that fold up and come together to make the three-dimensional shape of a finished protein? Okay, so a couple more concepts we've already talked about is the idea of steric hindrance, which limits the closeness that two amino acid residues can come together. So two large R groups are not as able to come as close together as two small R groups are. Okay, so another very important element in the way that polypeptide chains can fold up is how hydrophobic or hydrophilic the different amino acids are. So polar side chains, okay, so here is a polypeptide chain with its peptide linkages illustrated just as a white tube, if you like, with the R groups of different amino acids sticking out on either side. And some of these R groups are polar, they're charged, they're hydrophilic, they like to be in contact with water. Some R groups are not charged, they're hydrophobic, they're non-polar, and therefore they do not like to be in contact with water. So there is a great energetic pressure, if you like, an energetic advantage for this polypeptide chain to fold up in a way that the preferences of each of the amino acid side chains it's allowed to be in its preferred space. So the hydrophobic residues come together and are on the inside of the protein, while the hydrophilic residues are on the outside where they can make contact with water. Okay, so this is what happens in a watery environment, in an aqueous environment, and of course, physiological conditions, conditions inside cells, conditions inside the body is an aqueous environment, a water environment. So here are charged parts of the protein making contact with the water, allowing the protein to be dissolved in the water, and the hydrophobic amino acid, shown in green, folded away on the inside where they're making contact with each other and therefore not in contact with the water. And this is a very energetically stable shape, stable conformation for the polypeptide chain. Okay, all the amino acids are in their preferred environment. Okay, so polypeptide chains often fold because of these hydrophilic and hydrophobic interactions. Once they have adopted that folded shape, a lot of other interactions start joining in as well, which stabilize that folded shape and hold it in place. So here are a number of different interactions that can be important in protein structure formation. Very important is hydrogen bonds. I hope that you remember how a hydrogen bond forms. Hydrogen bonding is very important for interactions between different parts of the protein. And similarly, electrostatic interactions, attractions between positive and negative charges on different amino acid side groups. Okay, so each of these bonds, if you like, is a weak bond. They're non-covalent bonds, electrostatic bonds, hydrogen bonds, and also van der Waals attractions between large functional groups. These methyl groups here are attracted to each other and somehow forming a bend in the protein. Okay, so these come together by van der Waals attractions, and you can see that the protein here has adopted a folded shape. So non-covalent interactions are weak compared to the covalent bonds, but when there are many, many of them, they can have a big effect in stabilizing the folded structure of a protein. So all these things are acting together. The limits set by rotation and steric hindrance in the polypeptide chain, the hydrophobic and hydrophilic preferences of each of the amino acid residues, and the non-covalent interactions between different parts of the polypeptide chain. So these all work together to fold up the polypeptide chain into a folded protein. Okay, so a large number of weak forces acting together finally produce strong interactions that are enough to hold a protein in its folded shape in most conditions. So most proteins have a single most stable conformation, that's a word for the folded shape of the protein, under physiological conditions. So remember that things like pH or ionic strength can affect the charges on amino acid side groups, for instance. So a protein might have a different conformation at different pHs. But if we're talking about physiological conditions around neutral pH, there will be one most stable conformation for that particular protein. So our diagram here illustrates an unfolded polypeptide chain folding into areas of secondary structure, which then fold together to produce a tertiary structure. So we'll be following through what's going on here. Most proteins fold to their stable conformation spontaneously. They do it by themselves. So the sequence of amino acids in the polypeptide chain, the primary sequence of the protein, has all the information that's needed to produce the folded shape. The information is the nature of each of the amino acids and where they are in this polypeptide chain. And because of those particular amino acids being in the particular place they are, that is what gives the polypeptide the property to fold up into its correct conformation, the correct folded shape for that particular protein. Okay, so here is an illustration of how this can happen. If a protein is treated with urea, a high concentration of urea, that urea breaks the non-covalent interactions. It disrupts the nature of the interaction, the hydrogen bonding and so on. The protein loses its folded shape. It denatures, it loses its normal nature and becomes denatured. So here we can see a folded protein, a purified protein. Here we have placed it, we've dissolved it in a high concentration of urea, and all the interactions that are holding this polypeptide chain in its folded shape are broken by the urea, and we see an extended polypeptide chain, a denatured protein. Okay? But then if we remove the urea, the protein folds up again. It readopts its original conformation, because all the information for this folded shape is still there in the amino acid sequence. The only thing that stopped it being folded in this state is that the interactions that caused that folding were broken. Okay, so this shows that most proteins, or many proteins, don't actually need anything else apart from their own amino acid sequence to fold in the correct way. I've put a little note here. There are some proteins which need other proteins called protein chaperones to fold them. We'll talk more about that in your molecular biology course next semester. But most proteins, many proteins, are able to fold by themselves. They don't need a protein chaperone to fold them. Okay, so let's see what's happening as the protein folds. So here's the unstructured polypeptide chain again. And here we see secondary structure forming. We've got two sorts of secondary structure illustrated here. In blue, we have a helix, an alpha helix, and in green, these arrows represent a beta sheet. We'll be talking more about those in a minute. So these are elements, regions of secondary structure that have formed by interactions between nearby amino acids. So you can see that this helix is caused by amino acids which are actually next to each other in the polypeptide chain. Okay, so here's one, and here's another, and here's another. So each amino acid starts to interact with other ones which are close by to start forming that secondary structure. So let's look at some of those elements of secondary structure. So here's a definition of secondary structure. The local spatial arrangement of the main chain atoms in a segment of polypeptide chain. If you're a person who likes learning definitions, that's a good definition to learn. So two common types of secondary structure. There are a few more, but I'm going to be talking about alpha helix and beta sheet today. Okay. So pause for questions. A sip of water. Excuse me. Okay. So an alpha helix. Here is a diagram of a polypeptide chain folded up into an alpha helix. So if we follow along this chain, you can see the polypeptide backbone connected by the peptide bonds. It's going here, 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 and here. Okay. The side chains of the amino acids are sticking out on both sides of the backbone, and they're not actually involved in this formation of the helix. Okay, and then the helix is stabilized by hydrogen bonds here, 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 and so on. So this diagram takes away the R groups and the hydrogens that are sticking out in this diagram and just emphasizes the shape of the polypeptide backbone of the polypeptide chain. Okay, so here we're following the shape of the backbone as it twists in a helix shape. And the helix is just emphasized with this ribbon. So here we can see by the dimensions of it. This is about 0.54 nanometers, okay, between one turn of a helix and the next. And that is made up of 3.6 amino acids. So more than 3, but less than 4 amino acids, sort of about 3.5 amino acids to make one turn of the alpha helix of a protein chain. It's a right-handed helix. We'll be talking more about right and left-handed helixes when we do DNA and nucleic acids. But basically, the way to tell whether it's a right or left-handed helix is to look at your thumb and fingers, curve your fingers around and move your hand in the direction of the helix. If your fingers are curving in the same way as the helix, it's a right-handed helix. If it's your left hand that matches the direction, it's a left-handed helix. Okay, so this is a right-handed helix. The helix is curving sort of backwards here, forwards here, so it's curling to the right. I hope you can see that. Okay, so the hydrogen bonds are what holds the alpha helix in place. The hydrogen bonds form between the hydrogen atom that's attached to the nitrogen atom of a peptide linkage. Okay, so here is a nitrogen shown in blue, and here is its hydrogen, and here is an oxygen atom shown in red, and this is the oxygen atom of the carboxyl group of the 4th amino acid on the amino terminal end of that peptide bond. Okay, so here and here, here's one hydrogen bond between this hydrogen attached to the nitrogen and this oxygen attached to the carboxyl group. And another one here and another one here. It's a bit difficult to see, but if we follow round, so here is the first of the hydrogen bonds, and if we count, here's one R group on one amino acid, 2 R groups, 3 R groups, and then the 4th R group here is attached to the 4th amino acid. So 4 amino acids between the 2 involved in the hydrogen bond, and you can sort of count round and see for each of them. The other thing to notice is that the hydrogen bonds are parallel to the direction of the helix. Okay, they're not going across the direction of the helix, they're going up and down the helix, stabilizing that helix in its folded shape. Okay, so only the atoms of the polypeptide backbone are involved in forming the alpha helix. So it doesn't really matter which R groups are here for the sake of the formation of the alpha helix, but, or however, you see I've got a however there, different R groups can disrupt the formation of the alpha helix, because you can see here that in some places the R groups are brought quite close together, and it's necessary that the phi and psi bonds are rotated to allow this helix to form. So different amino acids have a different ability to form alpha helix. The amino acid that's most likely to form alpha helix is alanine, the second to smallest amino acid. It's a bit of a puzzle why it isn't glycine. It's almost as if glycine is too small. So alanine, whose R group is a methyl group, if this is polyalanine, this helix will certainly form, okay? This helix will very easily form. Other R groups become less likely to form a helix. So if we have quite a lot of alanines, but several of other amino acids, for instance, in a chain, it may or may not form a regular alpha helix. At the other end of the scale, proline, because of its imidazole ring structure, because its R group is joined to the polypeptide backbone, it's a lot less flexible than any of the other amino acids, and it is not able to take part in an alpha helix. So if there is a sequence of amino acids that are forming an alpha helix, and then there is a proline, proline will be the end of the alpha helix. Proline acts as a helix breaker. Okay. So pause for questions. Okay, so our next element of secondary structure is beta sheet. So here again, a similar diagram showing the polypeptide backbone and the R groups. And again, a diagram just emphasizing what the backbone is doing. So in a beta sheet, parts of polypeptide chain lie side by side, close together alongside each other, and hydrogen bonds form between the chains and stabilize the structure. Okay, so here we have one part of a polypeptide chain, another part, and a third part. And if you follow the directions of the arrows here, the direction of the arrow on the end of the chain points towards the C-terminal end of the polypeptide. So this one is going in one direction, opposite direction, and first direction again. Okay, do you see that? So hydrogen bonds, again stabilizing it, formed again between the nitrogen, or the hydrogen attached to the nitrogen on one chain, and the oxygen attached to the carboxyl group on the other chain. And this is an illustration of an anti-parallel beta sheet. Okay, so there are two different ways that beta sheets can form. This is what we've just seen, where there's a curving round, so a middle strand goes in the opposite direction. Okay, and then there could be another one here, and it could continue for some distance. This one is a parallel beta sheet, so each of the three chains go in the same direction, with a looping round of the polypeptide chain in between them. Okay, so both anti-parallel and parallel beta sheets are quite common types of secondary structure. So beta sheets, if you look at them from the side, okay, so this is a top view of what we've just seen. This is a side view. You can see that there's a sort of zigzag appearance with 7 angstroms or 0.7 nanometers between each zig and zag. Okay, and 6.5 on a parallel one here. Because of this, these are often called beta pleated sheets. Okay, pleated, the same word we use for a skirt, clothing, where the fabric is folded backwards and forwards to give it fullness. So a beta sheet or a beta pleated sheet mean the same thing, and both the anti-parallel and the parallel type of beta sheet both form this zigzag appearance from the side, although slightly different dimensions. Beta sheets can form between sections of polypeptide which are either close together on a chain or far apart, or can even be from different polypeptides coming together as part of making up a whole protein. It's different polypeptides joined together. Okay, any questions on beta sheets? Okay, so here is our folded protein again. So now you know that this is alpha helix. This is an anti-parallel beta sheet, and you can see here that the beta sheet is forming between two pieces of polypeptide chain which are quite close together. And again here, they're quite close together. So again, as this protein folds together, parts of this folded secondary structure come together to start forming the tertiary structure. So here we see the alpha helix lying on top of the beta sheet and interacting here. And then this happens more, another alpha helix lying over the beta sheet, and again, two alpha helices lying over this beta sheet. So two similar structures being formed as the protein folds up. And another folding brings together these two elements of structure into an interaction between them. Okay, an interaction between this part and this part sort of folded together to give the final tertiary structure of this folded protein. So again, at each stage, more and more bonds will be being formed as more and more amino acids, more and more parts of this structure find the correct place where they can find their most stable, most energetically stable conformation until we have the final folded tertiary structure. Okay, so a definition of tertiary structure: the three-dimensional conformation of a polypeptide in its native folded state. So native here means it's normal folded state. It's folded state that's found in nature. Okay, so here the diagram we keep looking at. So now you'll recognize that here we have an alpha helix. Here we have an anti-parallel beta sheet, and they're folded together with other parts of the protein which are in different structures but have not formed these regular secondary structures to give the final folded shape we're seeing here in this protein. Okay, so you'll see if you look at this diagram that amino acids which would be far apart if we stretched out the polypeptide chain into its linear form are being brought close together as the protein folds up. So if we look here, for instance, an amino acid that's part of this turn between the parts of a beta sheet is a long way away from an amino acid that's here on this pink part, okay? But when the polypeptide is folded up into this structure, these two parts actually come very close together. Okay, so if we follow around the chain, we have to go all around this pink part. Okay, and then how do we do it? Comes down here behind the thing and then into the alpha helix. So there's a lot of amino acids here around the alpha helix and down here onto the pink part before we get from this green turn to the pink part here. But in the folded structure, they're very close together. And again here, an amino acid on this bit. Again, it's quite a long way away on the lineage sequence, but close together in the folded shape. So this part of a protein might be very important. It might, for instance, be the active site of an enzyme. And there might be a number of amino acids that all form that active site. One here, 2, 3, and 4, for instance, which are on completely different parts of the polypeptide chain, separated by quite a lot of amino acids, but essential for the function of this particular enzyme. Okay, so correct tertiary structure, correct folding, bringing these essential amino acids together into exactly the right position in relation to each other is very important for the correct functioning of the protein. So even if it's only maybe 4 amino acids that are important to make the protein work, all these other parts of the protein are important for placing these 4 amino acids in exactly the correct places in relation to each other. So this is why correct folding, correct structure, correct tertiary structure is very important for a protein to work correctly, and why changing an amino acid, why a mutation can affect the functioning of a protein, even if it doesn't change one of the actual important amino acids that are essential for the function of a protein. Okay, so correct tertiary structure, usually extremely important, essential for correct functioning of the protein. Correct function depends on correct structure. Okay, so that's tertiary structure. And quaternary structure, here's a definition. The three-dimensional structure of a multi-subunit protein, particularly the manner in which the subunits fit together. So the important thing here is the word multi-subunit protein. So we'll be going on to learn a lot about hemoglobin, for instance. This is a representation of hemoglobin, and hemoglobin has 4 subunits, 2 alpha and 2 beta subunits, which come together and work together to make the whole molecule work correctly. Okay, so the quaternary structure is the way in which those subunits fit together, and also other things which are fitted into that protein. So this is the heme group. Okay, we'll talk more about that in a minute. So many large proteins are made from a number of smaller subunits, smaller proteins which have come together. And these might be the same as each other. They may be identical subunits, or they might be different subunits. So in hemoglobin, we see both. We see 2 alpha hemoglobin subunits and 2 beta hemoglobin subunits. So alpha and alpha are the same as each other, beta and beta are the same as each other, but alpha and beta are different from each other. Okay, so this is not quite the same as what I've already told you, that a single protein can also have several polypeptide chains. Although each of these subunits is a different polypeptide chain, if you disassociate them from each other, they still form a complete protein. Whereas in insulin, for instance, if you separate those 2 polypeptide chains, it falls apart and you no longer have a complete protein. Okay, so quaternary structure is about different subunits. So we've noticed that hemoglobin has something in it which is not made of amino acids. It's a prosthetic group. And in hemoglobin, it's a metalloprotein, a heme prosthetic group with an iron in it. Okay, you'll be hearing a lot more about hemoglobin, so don't worry about it, but it's just an example of a molecule with a prosthetic group, something which is not made of amino acids, but's attached to the protein. So it's a conjugated protein, and the attached group is called a prosthetic group. So a metalloprotein is one example. Another example would be a lipoprotein, which has a lipid part attached to it, or a glycoprotein, which has a sugar part attached to it. Okay, any questions on that? Okay, so as we finish, I thought I'd say a few words about looking at diagrams of proteins, about the different ways that you see protein molecules represented. So here are 4 different common types of representation of the same protein. You can see that they look somehow a bit similar, but they're also quite different. So let's look at these and understand what we're seeing. First of all, different colors. This does make them look quite pretty, but it's not just done to make them look pretty. Often rainbow colors, the colors of a rainbow, red, orange, yellow, Richard of York gave battle in vain. Red, orange, yellow, green, blue, indigo, violet. Okay, they are used to represent the polypeptide chain moving from the N-terminal end. Remember, we always read protein sequences from the N-terminal amino terminal to the C-terminal, the carboxyl terminal. So here, the violet is at the N-terminal end and the red is at the C-terminal end. So if you follow through, you can see violet changing to blue. Is it going here? Changing to green. Where does it change? Somewhere here. Changing to yellow here, to orange, and changing to red. Okay, so this helps you to see how the polypeptide chain is folded up. So you can see here physically the N-terminal end and the C-terminal end are quite close to each other, even though they're opposite ends of the polypeptide chain. And this coloring just shows you and emphasizes that fact. Okay, so this first diagram is what's called a backbone model. It shows the overall organization of the polypeptide chain. So here we've taken away the side groups. We're not saying anything about the R groups. And we're not even really emphasizing secondary structure, although perhaps you can guess that this is an alpha helix by just looking at the shape, but we're not really seeing very much else about the shape. We're looking at the way the polypeptide chain is organized. This is what's called a ribbon diagram, and you can see that parts of it are the same as the backbone model. These are the parts which are not taking part in secondary structure. However, the secondary structure parts are shown as ribbons. Okay, this is why it's called a ribbon diagram. So here we have a ribbon of alpha helix, and here another ribbon of alpha helix, and here we have a beta sheet, which is actually lying along the alpha helix. And here we have some more beta sheet here, an anti-parallel beta sheet here and here, and another bit of it here. And I think this one will be interacting with that one as well. So here we're emphasizing the secondary structure while still showing the parts that aren't part of secondary structure as a backbone. If you notice, the beta sheets have an arrow on the end. This helps us to know which direction the polypeptide chain is going in. The arrows are pointing towards the C-terminal end of the model, okay, of the polypeptide chain. Okay, the third type of diagram is called a wireframe diagram. And here we're seeing an illustration of the R groups as well. We're seeing the side groups being represented, sticking out from the main polypeptide chain. And you can see that it's a lot harder here to follow the direction of the polypeptide backbone. It's a lot more cluttered somehow, although the colors do help a little bit to help to guess which direction it's going in. But because you can see the shapes of the R groups, you can also start to see important things like which ones are on the outside and which ones are on the inside, and how big the different R groups are, and which particular amino acids are in which particular place. So you can see if some R groups are close to each other. For instance, we notice the C-terminal ends and the N-terminal end are close together down here. You can see that there are R groups on the amino acids near those ends which are interacting with each other and probably holding those ends of the polypeptide chain in place in the protein. Okay, so that's a wireframe diagram. And the final type is a space filling diagram. And here the polypeptide chain is almost hidden. You can't see the polypeptide backbone at all. But what you can see is the surface of the protein. So each of the atoms that make up this structure is sort of given its atomic radius, if you like. So each of the R groups is being represented with the size that it is. Okay, so here, for instance, we have a ring. This is probably tryptophan, I think, sticking up from the side. And here we see that represented. So we're seeing a large R group sticking out from the surface of the protein. Okay, so this can be important in thinking about how the overall structure is folded up and its relation with water and things like that, for instance, or what other molecules can come into it. Perhaps it interacts with a substrate and has a binding pocket. We'd be able to see that easily on this sort of diagram. Okay, so there are the four diagrams again: backbone diagram, ribbon diagram, wireframe diagram, and space filling diagram or model. Okay, so I want to encourage you to do some work for yourself now. I'd like you to explore some protein structures for yourself, and I'd like you to do that by going to one of the protein data banks I mentioned earlier. The American Protein Data Bank has an educational part, which is called PDB for Protein Data Bank 101. So I'm just going to take you online to the front page that you see. So if you go to that web address, this is what you'll see. Let's just move me to one side so you can see better. Okay, so this is the front page of PDB 101. Each month they highlight a particular molecule. This week it's a drug called cisplatin, which binds to DNA. You'll learn a bit more about that in the molecular biology course next semester. I'm very interested to see that this diagram is new on this website. But if you go to the Learn tab here, you can see lots of things here under Learn. Okay, so please do explore these things. There are all sorts of things, coloring books, posters, animations. If you go to videos, there is an excellent video called What is a Protein? So that summarizes really everything we've been talking about proteins in just a few minutes. So if you want to really cram your knowledge of proteins, that would be a good video to go to learn more about proteins. If I go back to the front page again, under the molecule of the month, you can find a number of different molecules, and there's a few particular ones that I'd like you to look at. So if you go to molecule of the month, you can arrange them by title, okay, and then you can just browse through alphabetically the different molecules that have been featured as molecule of the month. Okay, a lot of them begin with C for some reason. And one of the molecules that you can see here under C, whereas as it is collagen. Okay, so here is collagen. So here's what we see if we click on collagen. Here is a page all about collagen. Okay, so this is the molecule of the month page for collagen. You can see here information about collagen, and here is the structure of collagen. Some nice illustrations and some nice things to think about. And down here, exploring the structure, we have a static diagram, if you like, showing the different amino acids involved in collagen structure. But if you click on this link, and if your device that you're using is capable of it, you can actually get an interactive three-dimensional model of collagen. Okay, so here is collagen. If you go to this diagram, this model, you can do things like turn it round, look at it from different angles. You can zoom in and out. Okay, and by going down here, you can color the chain in different ways. So collagen actually has 3 chains, which you can read about it above, and you can color them different ways to see different important parts of that structure. Or you can show, it says here, show atoms, which gives you a space filling model, or you can show the wireframe model. So do have a go with that. See what you can learn about collagen for yourself. Next time we're going to be looking at structural proteins, and you're going to be learning about collagen. So this is a good way to learn about collagen. Two other molecules of the month that you might want to look at, we're going to be talking a lot more about as well, are myoglobin and hemoglobin. Okay, so look at those, read about them, and explore the protein database. There's a lot of good things in here. Do have a look at the other things here under learn. You can look under the health tab. Lots of interesting things here that you can explore. Okay, so if I go back to my PowerPoint now, wherever it's gone. Okay, it is underneath. Okay, so I hope that you're going to have time to do that. And I think that just exploring, just finding out what there is there, especially about collagen, myoglobin, and hemoglobin, will help you as we go forward in the next lectures. Any questions on that? If you have difficulty accessing the protein data bank, let me know. And finally, just to remind you where we are in the textbooks, if you're using the old edition of Leninger, you'll be wanting to look at chapter 6. If you have the 5th edition from 2008, proteins are covered over 3 chapters, 3, 4, and 5. And if you have this latest edition from 2017, the information that we've covered today is in chapter 4, which is called the 3-dimensional structure of proteins. So do remember to look at the textbook as well. Check what I'm saying. I might be giving you wrong information. If you find anything that I've got wrong, please do let me know. And please do start learning to work for yourself, not just relying on the lecture notes. Okay, so I think that's all we have for today. I'll see you again next time. Stop recording.